welcome to AP Help 101. So today I'm going to be talking about cell division, and this is another key idea that comes up in the AP Bio test a lot. So just a quick introduction about cell division. So the main functions of cell division are reproduction, repair, and growth. That's what it's mainly used for. And there are two types of reproduction, meiosis and mitosis. Now mitosis is when two identical daughter cells are created, and those are called diploid cells. And meiosis is when there are four different daughter cells, each with half the amount of chromosomes of the parent cells, and those are called haploid cells. Mitosis is usually just normal cells, while meiosis is sex cells like eggs and sperm. So the structure of the chromosome. We need a little bit about the chromosome for us to understand what happens in cell division. So it contains sister chromatids, which are exact copies of each other, and there's a structure called the centromere, which holds the two chromatids together. We have a kinetochore on the centromere, which connects the chromatid to the mitotic spindle. So let's talk about the cell cycle. It's a sequence of growth and development, and its timings and rate are crucial to normal growth. If your timings and your rate are off, if the cell cycle is off, you're not going to be growing normally. And there are factors that do limit the cell cycle, cell size and cell division, which is the volume to surface area ratio. So the ratio of cell volume to surface area. The area of the cell membrane increases as the square of its radius. It's just like basic math. The volume increases to the cube of the radius. So therefore we know that the volume increases faster than the area. And this volume to surface area ratio is really important for when cell growth starts and stops. Capacity of the nucleus. The nucleus must provide enough information for the cell to function. If there's not enough information, then the cell won't be able to function and it'll just die. So active cells are usually smaller. And then there are also larger active cells which contain two nuclei. So if the cell is large, it may need to have two nuclei for it to function completely. But if it's just a small cell, then it'll probably just have one nucleus and it'll probably be very active. So, phases of the cell cycle. There are 5 major phases. We have G1 phase, S phase, G2 phase, mitosis, and cytokinesis. And you may think, okay, wait, isn't mitosis one of the forms of cellular division? Yes, it is, but it's actually also one of the phases of the cell cycle, and I'll actually explain that more. So interphase. Interphase is also known as the G1 phase, S phase, and G2 phase, all put together. That's interphase. And most of your cells are in interphase. You know, for most of their growing period, they are in interphase, and then they move to mitosis and cytokinesis. So the G1 phase is where most of the cell's growth takes place. The S phase is when DNA replication takes place, and this is when the centrosome is duplicated. And then G2 is just a continuation of growth. More than 90% of a cell's growth time is actually spent in interphase. So that just shows you how short mitosis and cytokinesis is, actually. And interphase is very important for the growth of the cell. So mitosis. It's also known as the division of the nucleus, and it has 4 stages: prophase, metaphase, anaphase, and telophase. And basically, just really briefly, what happens is the nuclear membrane disintegrates, the chromosome strands condense and are able to be seen, and then the mitotic spindle forms. The chromosome then lines up, single file, in the center, and the chromosomes positioned they're at position at 2 ends of the cell. And there are spindle fibers connecting the kinetochore to the centromere. The centromere separates, pulling apart the 2 chromatids, and the chromosomes finally cluster at both ends of the cell. Condensed chromosomes unwind and 2 nucleoli form. And this is actually prophase, metaphase, anaphase, telophase in this order that I wrote it. It's prophase, metaphase, anaphase, telophase. you don't need to know what specifically happens in each stage. That's what they changed in the AP Bio test. But you do need to know the general gist of what goes on during mitosis. And finally, cytokinesis. It's just a division of the cytoplasm. Cytokinesis, kinesis, division, cytoplasm, cytokinesis. A cleavage furrow is created in the middle in the animal cells, where that's basically just a little hole there, which will separate the two cells out. In plant cells, that is actually called a cell plate, and it's creating down in the center. Plant cells are more rigid than animal cells, so they don't make cleavage furrow. They don't squeeze in, but it's just a straight plate. And the daughter cells don't separate in plant cells, actually. They don't separate in plant cells. So now we're going to talk about cell division and cancerous cells. Just briefly, just a little bit about this. Normal cells do divide until they become crowded. That's called contact inhibition, density-dependent inhibition. There's also something called anchorage dependence, which they will stop growing until there is a surface touching the cell, and then it stops growing. However, in cancerous cells, these inhibitions that usually stop cell growth don't work, and that's why they continuously grow and grow and grow until it becomes a problem and it turns into a cancer. So that's what's dangerous about cancerous cells. That one cancerous cell could result in a huge problem. So next week I'm going to talk about more on meiosis. This was just on mitosis. But next week I'm going to talk more on meiosis because it's a lot more different than mitosis is. This is just a brief summary as to what mitosis is. And meiosis is actually a completely different process. It has some similarities in it, but there's more steps to it, and there's more to explain in meiosis, so I'm going to discuss that later. If you have any questions or comments, leave them down below, hit the subscribe button, and I will see you guys next week.